Hello and welcome back to AP Bio. And today we're going to talk about Mendelian genetics. Before we get started, I thought I'd play this short biography video just so we can get an idea of who Gregor Mendel was. We always talk about how he's the father of genetics, but what did he do? What was his contribution? Why was it so important?
Monk, teacher, and biologist, Gregor Mendel is known today as the father of modern genetics.
Gregor Mendel's experiments on pea plants were fundamental in establishing the field of modern genetics.
Born Johann Mendel on July 22, 1822, in what was Austria and is now the Czech Republic, Mendel excelled at school and became an Augustinian monk to further his education. He was given the name Gregor when he entered the monastery.
The monastery sent him to the University in Vienna, and that gave him the education in science, math, chemistry, and botany that he then brought back to use in his teaching.
Mendel accepted a teaching position at the monastery in Brno after completing his university studies. It was during this period he began his famous pea plant experiments.
Before Mendel, people thought you would equally inherit the traits of parent A and parent B in equal proportions.
Mendel experimented with pea plants and found that instead there were dominant and recessive traits in all organisms that were passed on statistically based upon the mixing of genes between the two parents.
This theory is now known as the law of segregation. Mendel also formulated the law of independent assortment.
The law of independent assortment addressed the fact that there are a large number of traits. These traits are passed on randomly and are independent of the other traits and of the traits of the two different parents.
The ability to explain hereditary diseases and to engineer plants with desired properties are all consequences of the fundamental theory founded by Mendel.
During his life, Gregor Mendel's research was disregarded by almost everyone. It wasn't until the early 1900s, after his death, that Mendel's work was appreciated and now serves as the basis for genetics and heredity.
In his later years, Mendel became the abbot of his monastery and spent less time with his scientific endeavors. Gregor Mendel died in 1884 at the age of 62.
Gregor Mendel's work laid the foundation for modern genetics, for modern gene therapy, and is the basis for some of the most exciting discoveries in medicine currently and in the near future.
All right. Excuse me. So, move on to next slide, please. Thank you. Okay, so the video mentions the law of segregation, the law of independent assortment. And so we want to talk about what these laws mean. So the law of segregation, here's a quote from your textbook: two alleles for heritable characteristics segregate during gamete formation. Okay, well, that's all well and good. Let's put that in normal people language. So in other words, two alleles for characteristics separate during meiosis and end up in different gametes. Now remember, these alleles are on chromosomes, so it's the physical separation of the chromosome and those versions of the traits that go with it. And so all body cells have two copies of every chromosome, but during gamete formation, we're going to separate those homologous pairs. They're going to end up in different sex cells. So each egg and sperm is only going to get one of the two alleles present in body cells. Again, they're located on chromosomes, but those will physically separate from one another. And so the best way to show that is a monohybrid cross in a regular Punnett square. So let's just review the Punnett square. You might want to pause here for just a second and answer the questions, and then Avi, I'm going to show you the answers in just a moment. Okay, so question 1, where do offspring get their genes from? That's obviously the parents. Question 2, we have 23 chromosomes in our egg and sperm cells. Question 3, the dominant allele is always shown by a capital letter. In this case, it's capital A, Recessives are always shown by a lowercase letter, so lowercase a. Parent 1 is heterozygous, having both one dominant and one recessive allele. Parent 2 does have the same genotype as parent 1. And of course, since both parents have the same genotype, they're going to have the same phenotype. So again, the law says that during meiosis, the alleles located on chromosomes are going to segregate into separate gametes. So I really like this illustration because we can track these little sections here on each chromosome would represent a sequence of gene, and there are different versions or alleles. And so they will physically be separated into different sex cells. So up here, if you're homozygous dominant, you've got both copies of dominant alleles. Well, your egg or sperm are just going to have both dominant. So those are like your only options. Same thing if you're homozygous recessive. These are the only alleles you have, so that's the only option you have to pass on to your offspring. In a heterozygous situation, when you have one dominant and one recessive, half of the sex cells are going to get the dominant allele and half are going to get the recessive allele. So this is what we mean by segregation. So how does this relate to a monohybrid Punnett square? Well, when you were in middle school and freshman biology, you're always taught when you work a Punnett square to take the two letters and separate them out, one over this column and one over this column. And over here, one in front of this row and one in front of this row. And so when you did that, you were actually modeling segregation. I don't know if you ever taught that, but that's what's happening. So body cells have two copies. They're both heterozygous, but the gametes only get one copy, homozygous or, dominant or recessive. Then as you work the Punnett square, each box is a prediction if this sperm fertilizes this egg. So if these two gametes come together, you're going to wind up with a homozygous dominant. If it was this sperm fertilizing this egg, it would be big P, little p, same thing here. And finally, if this sperm fertilizes this egg, we wind up with two little p's. So it's just kind of predicting the genotypes of the offspring. And then finally, each box represents the mathematical expectation of offspring. So in this case, we would expect out of like all the offspring, 75% would have purple flowers and 25% will be white. That's the expectation. It doesn't always work out that way, which is something we're going to look at in later lessons. As for the law of independent assortment, this is where independent assortment kind of comes into play in metaphase 1 of meiosis 1. So again, quote from the text, each pair of alleles will segregate independently during gamete formation. Well, what does that mean in real people language? So each allele on a chromosome is independent of the others and will separate independently into gametes. So it's just a matter of where those homologous pairs line up at the metaphase plate as to which gamete they'll be sorted into. And so these alleles aren't linked to each other at this point. Well, again, some of this stuff's for later lessons. So the best way to show independent assortment, is through a dihybrid cross. So dihybrid cross is going to track two traits at one time. So in this case, we're going to look at pea plants that are tall and have purple flowers versus short pea plants that have white flowers. So the thing about Mendelian genetics, everything is either or. This is either tall or short. They'll either have purple flowers or they will have white flowers. So we're going to use big T for tall, little t for short, big P for purple, little p for white. And so remember, in meiosis, we start with this, the starting cell. We're going to replicate the DNA. We're going to separate the homologous pairs. Then we're going to separate the replications from one another. And we wind up with 4 sex cells total, 4 gametes total. So, what would be the genotype of a homozygous dominant? That would be 2 big T's, 2 big P's. Now, what's going to happen in the gametes? Well, remember, due to segregation, each sex cell is only going to get one letter from the pair. So that means that the gametes will have 1 big T and 1 little p, but we would need to use that combination 4 times in the dihybrid Punnett square because there are 4 sex cells total. All right, what about homozygous recessive? Well, obvi, those are going to be all little letters. And the gametes, you only have one option. You're going to get one from each pair. Since they are both recessive, all the gametes are going to be little t, little p, again, for a total of 4, because there are 4 sex cells that are created. All right, well, okay, what if you're not homozygous dominant or homozygous recessive? What if you have heterozygous, you're tall, but you're heterozygous, and you have purple flowers, homozygous dominant? So there are 2 options. You could have big T, big P, or little t, little p. I'm sorry, little t, big P, because each gamete is only going to get 1 from the pair. And so again, you would have 2 of these for a total of 4. I'm just abbreviating here by showing you 2. What if you're heterozygous for both traits? This is where it gets a little more complicated, but again, they sort out so that every possible gametic combination gets 1 allele. So you could have big T, big P, or little t, big P, or big T, little p, little t, little p. So there are 4 separate gametic possibilities. And full disclosure, predicting gametes is one of the more difficult aspects of this, but it's really not that bad. You're going to be fine. So the reason we have to show all 4 is because in a dihybrid cross, it's a 16 box Punnett square. So there are 16 possible offspring combinations. And so that's why I was saying earlier, so like if this one happens to be heterozygous, so we list all 4 gametic combinations across the top and all 4 along the side. If this was homozygous dominant for both traits, big S, big Y, then we would just use big S, big Y all the way across the top, because that's the only possibility that that particular genotype would have. But that's boring because it's the same for everything. These heterozygous ones are more fun. Okay, so now I'm going to give you a chance. So if you want to pause here and see if you can sort out the gametes, I've obviously given you some hints by covering up the number of boxes. See if you can give it a go. All right, so there's only one option for the first genotype, capital B, lowercase e. There are 2 options here. You can have big B, little e, or little b, little e. And then the heterozygous, this is where it gets fun. We've got 4 different possible combinations. You can have both the dominants. You can have big B, little e. You can have little b, big E, or both recessives. And again, if you were using these genotypes in a Punnett square, these gametes, they were all 4 across the top or down the side would be this option. Or if you only have 2 possibilities, you would repeat these. So you'd have this one, then you'd use big E, little e again, and then the 2 recessives. Okay, so now that we've gone through all that, let's look at it and do an application. So let's say we've got some guinea pigs and they are true breeding or purebred. So P is the parent generation. So they are true breeding because this black guinea pig is homozygous dominant for black fur and for having short hair. This one is homozygous recessive for white fur and long. And so when we cross these 2 guinea pigs, we don't even need to work a Punnett square for that one, because clearly every offspring is going to get 1 allele from each parent. They will all be heterozygous for both traits, but they will express the dominant phenotype. Even though they don't show this phenotype, they still carry the alleles for it and can pass it on to their subsequent generation. So all the F1s, the first filial generation, are heterozygous for both traits. Now, what happens if we cross 2 of those guinea pigs? We cross the F1s to get the F2 generation. So each box gets 4 alleles, 2 from each parent, and when you work the Punnett square, you want to keep the B's and E's together. So again, we filled across the top with gametes. So what's going to happen when this sperm fertilizes this egg? Now the chromosome number in the offspring are restored back to normal, and this offspring gets all dominant alleles. Okay, so we're going to start working our way down the chart this way. So that means that this guinea pig is going to be black and short. So will this one, and this one. So we see no changes from the parents as of yet. Next column. So again, black and short. Oh, wait, what happened here? So here we have a black and a long because the two recessive alleles combined in this offspring to give us a new phenotype that does not look like the parents. But again, the genes are there. They were just masked because the parents also had the dominant alleles. And so now we're back to the regular black short. There's another black long. Got black short again. Black short. Oh, what happened here? So look at this one. Now we have a white short, even though this offspring does not look like the parents. Remember, they carry the alleles for this combination. Here we have another white short. Then we're back to the regular ones that look like the parents. Here is another black long. Here we have a white long, and then finally the white long. Sorry, I may have misspoke. This one, you've got the pictures. You can see what color they are. I apologize if I said the wrong phenotype. But lo and behold, right here, we get one that doesn't look anything like the parents because of the way the sperm and egg came together to create this. So this phenotype actually skipped a generation. This would resemble grandma, if you will, grandma guinea pig. All right, so as we look at the ratio, you can go back and count. There are 9 black short, 3 black long, 3 white short, 1 white long. I would suggest you memorize this ratio. There is no reason to work a dihybrid Punnett square when both parents are heterozygous because it's always going to be 9-3 to 3-1 under simple Mendelian genetics. So we say 9 have both dominant traits, 3 will have 1 dominant, 1 recessive trait, 3 will have 1 recessive, 1 dominant, and then 1 will have both recessive traits. So it's always going to work out that way. All right, now on your Google Doc, you've got some individual practice. Now, unfortunately, neither practice crosses are as easy as the 9-3 to 3-1 ratio. So you're going to have to work out these Punnett squares, but it's the only time I'm going to ask you to do this. So good luck and email me if you have questions.